Okay, hello. This is lecture 7, the one that was missed because of my migraine. So I am going to just record it, and there were some clicker questions, which I will put onto Canvas for you to do if you wish to. If you choose to do them, they will function kind of like a bonus towards your clicker mark. And yeah, so that should be a helpful thing. And so I'll start off by saying that this isn't an area of expertise for me, this topic. So I'm just going to be giving you a very general introduction. But sometimes people from 357 do end up working in the Matson lab. And that is the place to find further, much more detailed information. Basically, the project you will be contributing towards is a project that is meant to transform how rice does photosynthesis to try to make it more productive at higher temperatures. And we'll talk about kind of, why higher temperature is kind of bad for photosynthesis. And then we'll talk a little bit about the oligos you're going to design, the kinds of genes you're going to be targeting, and so on. And then on Monday, we actually will be going over the process of, how to find the sequences for the oligos and so on. And you'll be working with them through the rest of the semester in the lab, starting, I think, around the Thanksgiving time. Approximately, that's where we'll be starting to work on it in little steps each week. And at the end of the semester, you'll know whether you have the correct sequences, and we'll put them aside, and the Matson lab can take them if they wish. Okay, so basically, these are the things that I'm hoping you'll be able to explain. The kinds of genes we're targeting, why we chose them. Oops, there's the first clicker question. Why we're using Ceteria italica instead of rice as our model organism. The C3, C4 situation. So there's one type of photosynthesis called C3, another type C4. C4 avoids the problem of higher temperature. So we'll go through that a little bit, just a little bit. As I said, this is not sort of like my top area of expertise. Sort of the overall aim of what the project is trying to do, and a little bit about the part that the Matson lab plays, and then why we're using the CRISPR approach that we are. So there's the non-homologous end joining, or the homology-dependent repair method, and we're choosing this one, and there's a reason for that. So these are the kinds of questions you should be prepared to answer about this project. Okay, so that's the first clicker question. I will change it slightly since nobody was there this morning. So the background here is that in the United Nations, the Food and Agriculture Organization has determined that given population growth and so on, compared to, I think this data was collected in 2019, if I remember correctly, or 2018, that there must be a 70% increase in food production by the year 2050 to feed the world population. This is not taking into account things like global warming and so on. But here's some additional factors that sort of tie into this. We're seeing huge increases in food prices worldwide, which means that with our increased world population and the large levels of poverty, there are people who cannot afford these prices. Overall, worldwide, there is a somewhat increased standard of living. So people who are, who eat meat, are expecting to be able to eat meat more regularly than usual. And meat in particular uses up a huge amount of land, and there's massive amounts of energy input compared to the energy output for that. And there's limited opportunities for the expansion of agriculture. Now, one of the tiny possible silver linings of global warming is that some areas that are warming may not be under permafrost conditions anymore. So there may be a little bit of expansion that can take place. But most of the land that's that can be cultivated is currently being cultivated. So it's not like we can just find giant prairies where we can start growing a lot more wheat or something like that. And these days, there was the Green Revolution, which some of you may have heard about, where different kinds of fertilizers and different kinds of genetically engineered crops and so on increased yields and so on. But those returns have kind of happened, and more is going to be needed, and it's not clear how much more we can actually do about that. And of course, as I said, global warming. Photosynthesis has a temperature curve, and as temperatures rise, certain kinds of photosynthesis get much less efficient. If your plant can't do photosynthesis very well, it's going to produce a very poor yield. And so the question is, how do we stop that problem with yield as temperatures increase? And this is just a little bit of stuff that I got from the Food and Agriculture Organization. The United Nations has tons of interesting graphics and so on about this kind of topic. And I'm just going to provide some links because I think some people may be really interested in this kind of work. It's the kind of work that could really make a difference to people and to their lives. And if I was a younger person, this is exactly the kind of thing that I would think, okay, I'm going to build my career in this kind of area. And so this is just a projection, and notice it's a little bit out of date, but I couldn't find a more updated one. However, so this is the number of hungry people. So we're talking, this is in millions. So this is a billion. Oh, for heaven's sake. Starting to wonder there's something wrong with me. So this is a billion, right? So we're, this is a lot of hungry people. And this is kind of where we want to go. And this was when a lot of new technologies came into place for agriculture. These are projected, right? This is what was thought was going to happen. Right. But this is what really happened, and this is a projected one. This is not, we don't know this for sure, right? But this is still the target. So the United Nations and the people that are focused on world hunger, this is what we're trying to see. This is what they want to see, and so the kind of project that we're going to be talking about. Of course, it has to, it's going to have to start up higher and, start trending down. But this is the plan. This is the aim. This is what is being attempted by all the different groups that are working in this field directly. These are just some pieces of information that I pulled off the United Nations site. They have 17 sustainability goals. Some of them are very interesting. You might be, interested in taking a look, things about equality of access to education and so on. Food security, etc. But this is the one that our project is most connected to, the idea of ending hunger, achieving food security and nutrition, and promoting sustainable agriculture. So what we want is agriculture that doesn't over and over and over deplete the environment until there's nothing left, to grow more crops. You want sustainable systems so that whatever you put in, you get back out, and it keeps renewing, right? That's the kind of the idea. This is just a map of the mean temperature change. It's important to note that there isn't any place where it's actually gone down. These are the decreases, and these are the increases here. And this tells you approximately how many degrees the average temperature has changed. The ones that are clear are just ones for which no data have been provided. So it's not that nothing has happened there. It's just nobody has measured it and reported it. Okay. And there's a disclaimer down here. If you have any political leanings in some of these boundaries you don't agree with, that, this is just kind of based on reported data. So they're not making any claims. They're just, that's how the data were reported to them. And then this is the one that, is really awful. In 2023, 2.3 billion people had to struggle to be able to have adequate food on a regular basis. And 864 million had severe food insecurity, which means these are the people who are actually starving. You know, these people are malnourished. These are starving. Both unacceptable, but you know, this one is really, really bad. And then this is a little bit old as well, but it basically shows, like, if you look per year, different time periods, the amount, it's not that the yields are not increasing anymore, but the proportion of increase, the amount of increase is dropping. This is what I meant when I talked about diminishing returns. So that in the 60s up to the 90s, which is when a lot of our green revolution technology, the new pesticides, new not herbicides, but fertilizers, and some beginnings of genetic modifications and so on, you would see these really big increases in yield. So like here in wheat, a 3% per year increase, 2.5% per year in maize, and so on. And then rice and soybeans. These are our main staple crops that are followed. But if you look in the 17, excuse me, following that, the amount has decreased, right? And they don't have, at this point, they didn't have data for this, except basically there was less labor and less land available. So the idea is the labor, people who work in agriculture is not increasing by as much as it used to, and the available land is not increasing by as much as it used to. Currently, the available land is hardly increasing at all because it's mostly cultivated. So this is kind of background, the situation, to the best of my understanding of it, based on just doodling around in the United Nations websites and some of the other places, the institutes and organizations that cover these kinds of things. So I want to introduce now the reason why photosynthesis is problematic. And that is because in organisms that photosynthesize, there is an enzyme, and we call it Robisco, and it's ribulose biphosphate carboxylase oxygenase. This is an enzyme that can modify the starting material. So this is a C5, a 5-carbon protein, or not protein, carbohydrate, sorry. And the enzyme can either act as an oxygenase and modify. I'm not expecting you to memorize these. I haven't memorized them myself. But it makes a certain molecule whose name you don't need to remember, but it's a C, 1, 2, 3, 4. It's a C5, and it's been oxygenated. And then when this gets broken down, it makes a C3 molecule, 3-phosphoglycerate, which you could remember. You may have actually studied it in other contexts. But there's only 2 carbons here. This is 2-phosphoglycolate. And this actually goes through photorespiration. So in normal photosynthesis, which is down here, if it acts as a carboxylase, it incorporates this CO2. And that basically then hydrolyzes it into 2 C3 molecules that have the same chemical structure. And both of these feed into photosynthesis. So if the enzyme does this, the oxygenase part, only half of the molecules that are generated, you have basically half the productivity. And that means you need a lot more photosynthesis to build the same sugar molecules that are then going to build the body of the plant and allow for it to, in the case of rice, produce lots of rice grains. Now, some of the factors that affect the choice of the substrate, what's helpful is that it has a much greater affinity to carbon dioxide than to oxygen, 100 times more. But there's a lot more oxygen in the atmosphere. And then, so CO2 is not as present in the air. And then the thing that's not really kind of given a number here, but water solubility is really important because the gases move inside the plant through the stomata, but then in order to participate in the reactions that are going to take place, they have to dissolve in water, and then the enzymatic reactions are taking place in the cytoplasm, right, the cytosol. And so basically, under normal circumstances, just despite this huge difference, and this isn't as huge, but it's still an issue, we still have a threefold increase in the use of CO2 over oxygen. So there is still some use of oxygen in a normal plant, but most of the time we're using carbon dioxide, and therefore the plant is happy, it's making enough sugars and so on. However, as temperature increases, the solubility in water of carbon dioxide decreases a lot more than oxygen does. They both are less soluble at higher temperatures, but we would still call this in the sort of medium range for oxygen, whereas it's much lower for carbon dioxide. And so basically, overall, at the higher temperature, the carbon dioxide solubility decreases more than O2. And so what we see is we'll see a big increase in the use of oxygen as the substrate, and then that's going to drop the photosynthetic output, right? And so this is the crux of the problem with temperature increases. So, but there are some plants that they can avoid the photorespiration, and they do this through both some alterations in biochemistry that we're not really going to talk about in a lot of detail, of course, because it's not really my area. I see a typo, darn it! I always see the typos too late. But they basically, instead of feeding C3 molecules into the process, they start with C4 molecules, so 4 carbons. And they have this special anatomy where the photosynthesis takes place in a different location from the other types of plants. There's special cells. And so basically what happens is the CO2 is captured in one type of cell, and then it's transported by a truly little shuttle system that picks up the CO2 and kind of delivers it into the bundle sheath cells and creates that C4 carbon compound. And what that means is that the CO2 is being concentrated in there. Even if its solubility is low, it's picked up and dragged into these special cells where now the concentration is going to increase. And there isn't much access to O2 except what diffuses naturally into those cells. And so therefore, it's not used very much, if at all, as a substrate for this enzyme. So that's going to not maybe always make perfect sense, but I have some pictures coming up which should help a little bit. Okay, so basically, just ignore these plants. These are like very tolerant of arid environments and so on. They're succulents and cacti and things like that. So if we look at C3 plants, this is kind of the potential range of the output of photosynthesis. So the best temperature is, looks like it's about 26, right? And that's where you get your kind of optimal output. C4 plants, though, their optimal range is kind of in this area here. So it's more like, I think this line is slightly offset. It should be sort of right there in the center. So we're talking more like 35, 36. And it would be really great. And the idea of the project I'm going to try to describe, not in a lot of detail, but just enough for you to kind of understand what we're talking about, is that the idea is to try to move rice into this region so that if temperatures increase a lot, we will still see good productivity. And if you look at the photosynthetic rate here, even if the temperatures don't increase very much, suppose it stopped at a certain point, you're still going to have like more productivity, right? It's just for crops, productivity is what we're most interested in. So this is just a kind of a picture, just kind of show that. And oh, just to also note too that leaf temperature isn't the same necessarily as the air temperature, because the leaves are, the sun is blazing down on them, so the leaf temperatures are often much higher than the surrounding air. So that's just something to keep in mind as well. And so this is my sort of little schematic. I got it from here, and it looks like I might have not correctly copied it. But so in a C3 plant, so here's the stomata, and this is where your carbon dioxide comes in. And the cells in here are called mesophyll cells, spongy mesophyll. So I'll show you a picture in a minute, but they're kind of arranged in sort of bunches kind of around there with lots of gas exchange spaces in between. So the CO2 comes in and it can move all through, diffuse all through these little air spaces. But at some point it diffuses into the cell, it gets picked up by that C5 carbohydrate that I was showing before. It goes through a few steps that you don't have to know about, and 2 C3 are formed. This doesn't include the oxygen part. Remember that if oxygen gets in here, then basically you're going to end up with just one C3 molecule to feed into photosynthesis. And the CO2 can diffuse around, and it can enter those cells simply by diffusion. However, in a C4 plant, the carbon dioxide comes in. Again, it does have to diffuse into the cell. But then Pep-C, that I'm blanking on the name of, basically once the carbon dioxide is solid in solution, it becomes a carbonate ion, which again you don't really need to know. But so there's a C3 compound, and then the carbonate is added to it to make a C4, and then the C4 is actually transported directly into the cells next door, which are the bundle sheath cells. Okay, and what they do is they go, oh, okay, so they take the CO2 off, but then they feed that C3, or sorry, they take the CO2 off, combine it with the C5. The rest of this is exactly the same as that. The difference is what you're doing is you've got this little C3 molecule, whose, I wish I could just remember the darn name of it. This is phosphoenol pyruvate carbonate, and so this must be phosphoenol pyruvate, I think is the name of the molecule. It keeps picking up a CO2, and then it gets transported in here and it delivers the CO2, and then you've got the C3 back again. So it really is like a little bus system that brings that CO2 molecule into, and then allows it to be concentrated in here. And there may be oxygen around, but the CO2 can outcompete it because there's this whole mechanism of drawing it in and concentrating it in these cells, whereas whatever oxygen gets in here through just diffusing across the membranes of the cells. And this is just a picture. So this is wheat. This is a cross section. I'm just, it's not very nicely labeled, but here is the vein, the conductive cells in here. This is where the sugars and starches and so on are moved around. And the bundle sheath cells are sort of a structural feature, so they're kind of supportive, and that's this bit around here. Those are your bundle sheath. And then these are all your what we call spongy mesophyll cells. These are where all the photosynthesis is taking place. And this is a stoma here. See the little opening? They can open and close to let the gas in. And the same here. This is a stoma. And so sugars are being made in these cells. And then it has to be transported in here and then sent to different parts of the plants. Okay, so that's the anatomy of a C3 plant. The anatomy of a C4 plant, I think this is maize, if I remember correctly. You've still got the stoma, you've still got the air spaces, and you do have mesophyll cells that do have some chloroplasts in them. But the key difference is if you look at these bundle sheath cells, look at all the chloroplasts that are in here, and they're mostly kind of on the outside, not the outside the outside relative to the vein, the conductive cell. Because what's happening is that CO2 is being captured in these cells, and then it's being transferred, shuttled into these cells where the photosynthesis takes place, and then the sugar is sent here. And you're hugely concentrating the CO2 in this, and here's another one. This is the edge of another set of bundle sheath cells that surrounds the vein and so on. So this is referred to as the Krantz anatomy. It's just a different way of arranging not the cells, but exactly where the main amount of photosynthesis takes place, and it includes the Pep-C kind of shuttle that picks up the CO2 and then transports it directly and then lets it go in the bundle sheath where the photosynthesis is taking place. I hope that was clear. Okay, another clicker question. I will I will modify it for the Canvas. So there's this huge project because people are worried. Rice is probably the biggest staple crop in the entire globe, and so there's a lot of people who are part of a working group that would like to be able to engineer rice to be able to perform C4 photosynthesis. And if you can't make it a complete C4 plant, at least to modify it in ways that will help to make it more productive at those higher temperatures. There's a huge number of changes that have to take place to make the full transformation. But if you can even make a partial transformation, it's helpful. And so you'd need a lot of changes. You'd need some biochemical, so you can easily introduce enzymes to do a particular job, but they also have to have the right structures and anatomy in order to do them. And there are a number of different groups, and the members are focusing on different aspects. So the SFU lab, Jim Matson's lab, they're focused on the development of increased vein density. More veins and closer together, because that is one of the key, most important changes that needs to take place in order for the other changes to happen. And I'll show you some in a minute, some evolutionary data that shows how we can know what the order of the changes are. And so it was suggested that you could, if you could actually alter the photosynthesis that rice do, you could increase the output by 50%, which is not a trivial amount considering what a large crop it is. Okay, another clicker question. So what makes people hopeful that this can be done is that if you look at plant phylogeny, C4 photosynthesis evolved from a C3 ancestor many different times. So if you look here, you know, it happened here, it happened here, it happened here. Anywhere where there's a black line that branches off into blue, it happened here. It's at least once here, and then this, I think, is just one event, but there's a couple of events here, one here, and then another one sort of up in here somewhere. So the idea, oh, there it is, there's the other one. So it's evolved independently a number of times, and this is super helpful, partly because some of the plants that are currently existing do complete C4 photosynthesis. They've got all the features, but there are some that are only partially there. Remember that evolution is still in progress. Like, things are continuing to change. It's not like it's done. It feels done because we're looking around and we see, you know, what things are like now. But some plants are not finished the photosynthesis in that direction. And so we can look at plants that have a subset, just some of the features that we've observed in C4 plants, and see if we can identify an order in which the changes should be made. And it turns out that you can. So if you look at the different kinds of plants, you will find that the ones that are you know, very just barely C4, they have what's called preconditioning. So they have veins that are really close together. Okay, and there are more of them in the leaf by definition, because if the leaf is the same size and the veins are closer together, you have to have more veins in there. And so this is kind of like the first step. This is where the Matson lab is working. And then you need bundle sheath cells to be enlarged, and you need more organelles. That means more mitochondria and chloroplasts. I don't know why I'm blanking on that. And mitochondria have to be relocalized to a different position, the inner wall, because that's where they're needed. And then once all that is done, we have what's called the proto-cranial. So the morphology, the anatomy that is required, is basically set for the rest of the changes. And then there are some other things. There's like a CO2 pump, and there's increasing the sizes of the mitochondria and so on. And now you start seeing some of these enzymes. This decarboxylase has to be put in the bundle sheath, because this is the one that in the bundle sheath takes that CO2 off and kind of helps it feed into the photosynthesis. PEP-C activity, that's the one that puts the CO2 on the PEP molecule. Establishment of that cycle, and then optimization. So these are, and this just shows you the different plants and the fact that they, each of them have, these guys have just this feature, these guys have this feature plus this, these ones have all three, and so on. You don't find plants that have this without all these other things. This is how scientists realize that there are these steps that things happened, and you don't skip steps. You've got to have the anatomy before you can start messing with the enzymes and bringing them in, because they won't have any place to do the job that they're supposed to be doing. Hopefully that made sense. I feel like I'm blathering just a bit. Okay, and so just in 2010, a lot of the work really got underway. And this is an international group. These are people from all over the world that got together. Where was this? Paris, I think, or someplace, somewhere in Europe anyway. And SFU, there's Dr. Matson there, and maybe some of his students might be around here too. I'm not sure. But these were this is the beginning of this project, and each of the PIs here, their labs are taking a different part of the problem and trying to figure out how to make the changes. Okay, so I'm going to stop here because I suspect people are getting tired. A lot of new information. I will make a second one that talks more about like what we're going to do in the class and what kind of genes and so on. So there will be a second recording shortly.